My name is Dukundimana Paul, a student of Ines Ruhengeri, Biomedical, Year 3. We give an assignment regarding to presentation of the applied immunology. I'm a student of group 2. My task is from question number 26 up to 30. So it's time to start. Everyone who understanding this video, I request him or her to follow. Question number 26, it is asking, what is the significance of monoclonal antibodies in immunoassays? Monoclonal antibody are laboratory-made antibodies that recognize and bind a specific antigen. These monoclonal antibodies are important. They are produced by cloning a single B cell that produces the antibody of interest. Their significance in immunoassay are includes we are having a five. Number one, it's all about high specificity. Monoclonal antibodies are highly specific to single epitope on antigen, to mean that monoclonal antibody is targeting only one epitope of antigen, reducing background noise and empowering accuracy of immunoassays. Role number two, consistency quality. Because they are cloned from a single cell, B cell, monoclonal antibodies are uniform and they can be produced in large quantity, ensuring consistency and quality and, and reproducing across tests. Importance number three is versatility. Monoclonal antibodies can be used in a variety of immunoassays, including ELISA, enzyme-linked immunosorbents (ESI). Not only this, Western blotting, immunohistochemistry, and immunoprecipitation. Another role of this monoclonal antibody is diagnostic and, and therapeutic use. Monoclonal antibody are crucial in diagnosis for detecting pathogens, proteins, and biomarkers in a disease such as cancer, autoimmune disease, and infectious diseases. So those monoclonal antibodies, they are targeting those antigens and then helping the treatment. They are also used in the therapy such as targeted cancer treatment. Example, there is Herceptin for HER2 positive breast cancer. So another role, sensitivity and detection. In immunoassay, monoclonal antibody can enhance the sensitivity of the test by amplifying the antigen-antibody reaction, making detection of lower level antigen more reliable because it is highly specific. The next question, it is question number 27. How does immunoprecipitation work and what is its application? So here there are some answers on this question. Immunoprecipitation, first of all, is a technique used to isolate a specific antigen or protein from a complex mixture using antibodies. This process works as flow. How they work, we are having four steps in which this immunoprecipitation work. Number one is it is binding antibody to the antigen. A specific antibody is added to a solution containing antigen of interest. The antibody binds antigen to form antigen-antibody complex. Next, precipitation of complex. So it means that here, antigen and antibody has been reacted or coupled. Step number two, the antibody-antigen complex is captured by attacking the antibodies to insoluble support. Example, protein A or G bleeds, magnetic bleeds. This make the complex insoluble and allow it to be precipitated or pulled out of solution. Another step of making immunoprecipitation, it is separation. The precipitated complex, it is separated from the rest of solution by centrifugation or magnetic separation. The final step it is analysis. After precipitation, the complex should be analyzed by using technique such as Western blotting, mass spectrometer, or ELISA enzyme-linked reaction to identify antigen or study its interaction with other molecules. So now I hope everyone knows how to how immunoprecipitation work. Let me see application. In the applications, protein interaction. Identify and studying interaction between different proteins. Purification of the antigens by isolating specific antigen from further analysis used in research, and the characteristics of antigen complexity. Investigating the structure and the molecular weight of those antigens. All of those are the importance of immunoprecipitation. This was question number 27. Question number 28, here it is saying like what is immunocytochemistry and how it is performed. In this question, let me start by defining immunocytochemistry. It is a laboratory technique used to detect specific proteins or antigens in the cells using antibodies conjugated with detectable marker, typically enzymes or fluorescence dye. So it means that this immunocytochemistry, it is a technique we are using a specific antiprotein or antigens in the cells by using conjugated antibody with the marker or detectable and then typical enzyme or fluorescence dye. The process includes preparation of the data cells we are going to use. Cells are captured and then fixed into in the slide using chemicals like formaldehyde to preserve their structure and the proteins. Number two is blocking non-specific binding to prevent the unnecessary binding. Blocking solution is applied to reduce non-specific antibody binding, improving the specificity of antigen antibody interaction. Now next, we apply primary antibody incubation, which is specific to that protein or antigen of interest is applied to the cells. The antibody bind target antigen. Number four next, it means secondary antibody now which is labeled, which is conjugated, is now added to recognize that primary antibody. So secondary antibody that recognizes and binds the primary antibody is applied. Secondary antibody is conjugated to detectable marker, either enzyme, which is called Harish, Harish peroxidase, or immunofluorescence dye. So at the level of detection, after incubation, cells are washed to remove excess antibodies, and the signal from the marker is detected using immunofluorescence microscopy if we use fluorescence dye, and the light microscopy if we use HRP, which is enzyme. So this is the end of the question number 28. Let you go to question number 29. Question number 29, it says like, how does indirect fluorescence antibody testing work? So this is another technique. Indirect fluorescence antibodies or IFA testing is a method used to detect a specific antibodies in a particular serum, primarily for diagnosis infection or autoimmune disease. It means this is also techniques we are using in the diagnosis of disease, where we are using a specific antibody in patient serum. Yes. So there are steps to do in indirect immunofluorescence antibody. Step number one, antigen preparation. Antigen, virus or bacterium is fixed on the slide. Patient serum application means here, patient serum is added on that slide containing antigen. If the serum contains a specific antibody to against antigen, they will bind to it. Number three, secondary antibody incubation. After the washing off and bound antibodies, fluorescein rabid antibody, secondary antibody or which is conjugated. This binds to the patient antibodies, it is rabid. This secondary antibody, it is now conjugated with fluorescence dye. Next step, which is final, is detection. In the detection, thread is examined under fluorescence microscopy. If the antigen specific antibody present in the serum, the antigen will be fluorescence allowing the detection. Means by using this fluorescence microscopy, we can see if that complex has been detected. Final question, it is question number 30, saying that what is the flow cytometry and how is used in immunology? Are you interested? Yes, welcome. So flow cytometry, it is technique used to measure and analyze the multiple physical characteristics of individual cells in the fluid stream. It is widely used in immunology for cell sorting and cell analysis. The principle of this flow cytometry is this. Cells are suspended in the fluid and passed through the laser beam. The light is scattered by the cells and the emitted fluorescence from the labeled antibody provide detailed information on the size, granularity, fluorescence marker of individual cells. Fluorescent labeling. The cells are typically stained with fluorescently labeled antibody specific to structure, surface, or in intracellular marker, maybe CDs, cluster of differentiation, or cytokines. Step number 3, it is data collection. Means flow cytometry measure forward scatter, means cell size, and size scatter, means granularity and complexity of that cell. And fluorescence emitted from each cell will correspond to specific marker. Then final step is data analysis. Now means this data of flow cytometry is analyzed to quantify, means the number of the cells expressing certain. So, like conclusion, I have presented 5 questions. So I hope this answers is clear. Thank you. This is to take end of my presentation. My name is Tukunimana Paul. Thank you.